Alrighty, welcome everyone to our monthly Science of Alt Protein seminar. Super excited today to be joined by Dr. Peter Stogios, who is a senior research associate at the University of Toronto and GFI grantee. We've been thrilled to have him be a part of our grantee community for the last couple of years and are so excited today to learn about his research into how to produce low-cost growth factors for serum-free media. We will get started in just a few moments, but for those of you who are interfacing with GFI for the very first time, welcome, welcome. My name is Amy Huang. I'm GFI's University Innovation Manager, and I run our university level engagement, working closely with researchers and university administrators and students across the world to build scientific and training ecosystems. So if there's anything I can ever support you with, do feel free to let me know. With that said, I'm delighted to introduce Dr. Peter Stogios. As many of you know, a significant hurdle to the cost parity, to achieving cost parity of cultivated meat, is the cost of growth factors, which are really essential components of serum-free medium for sustaining cell growth, which is why we're so excited about Dr. Peter Stogios' research. And today he'll be diving into the exploration around how to lower the cost of growth factors in serum-free medium by more than 20-fold, by engineering the recombinant production of multiple growth factor families, and discovering new growth factors from exotic species that outperform the kind of conventional growth factors in sustaining multiple cell type growth. So lots of exciting information that I'm sure we'll have the pleasure of learning today. Peter, it is absolutely a joy to have you here with us. Again, we're so delighted to have you be a part of our grantee ecosystem and really appreciate you sharing your wisdom and knowledge with us. So whenever you're ready, do feel free to share your screen and we'll go ahead and get started.
Great, thanks very much, Amy, and thank you very much for the introduction and for inviting me here today, and Renee as well. Thank you so much. And just to the GFI in general, you guys are instrumental for this ecosystem and for transforming the industry. So thank you very much for everything you do. Just want to say that off the top. And I will share my screen. Okay. See my screen? Just want to make sure. Okay, great. So yeah, thanks a lot again for that introduction. I'm super excited to be here. So yeah, my name's Peter Stogios. I'm Senior Research Associate at BioZone, which is a Center for Applied Bioscience and Bioengineering at the University of Toronto. And we've been involved in this field for a few years now, but it was really catalyzed by our grant that we received a few years back from the Good Food Institute. And just by way of background, I'm a structural biologist and biochemist by training. And I think people like us, in terms of people that understand recombinant proteins, protein biology, protein function, play one key piece in this growing ecosystem and growing industry, but not, of course, not the only piece. So that's something really exciting that I've kind of encountered during this time that I've been in this space, is just the diversity of scientific backgrounds, and obviously economists and marketers, and it's just a really diverse community, and that's pretty exciting. I've learned quite a bit as we've applied what we know about protein biochemistry into this field. So the title of my talk today is Low Cost Growth Factors for Serum-Free Media. Let me just hide this little thing. Okay, and all the data that I'll be presenting today is published in a bioRxiv paper that we dropped a few months back. It's called Recombinant Production of Growth Factors for Application in Cell Culture. Or you can just go to bioRxiv and look for my name there and you'll find the paper. And the paper is also under peer review right now, so we hope to have it published in a peer-reviewed journal in the coming weeks or months. Hopefully only a couple months. So as we know, animal agriculture is hugely intensive, resource intensive. Obviously, animal agriculture has sustained the growth of human civilization for centuries, But increasingly, we know it's becoming unsustainable for a variety of reasons. So not only, is it ethical and concerns about animals and the way they're housed in factory farms, but also from the perspective of greenhouse gas emissions. So on the bottom left, I found this plot. It's a little bit dated now from 2014, but it's from the IPCC. I don't imagine the percentages have changed that much. But regardless, at that time, it was estimated that 24% of worldwide GHG emissions were from this bucket of agriculture, forestry, and land use. Obviously, agriculture will be a subset of that 24, but it goes without saying that significant greenhouse gas emissions come just from producing food protein from animals. That's very clear, and obviously we want to reduce that. Also, and this kind of blew me away when I found this map, just the land usage for raising food animals, as much as 40% of US land, and this is just a kind of a cool graphical visualization of that I found from Bloomberg, 40% of land is used just for raise food animals. To me, that's not really sustainable. We could be using that land for other productive purposes. As well, other resources like water use, electricity use, antibiotics, and I do a lot of research in this field. We know that just to sustain the health of animals in such environments, antibiotics are necessary for that, but that breeds antibiotic-resistant bacteria that can then transfer through the food chain to humans and cause disease. As well, viruses or zoonotic infections are known to transmit from food animals. COVID-19 is known to have transferred from some animal, we don't know which, but likely a food animal in the Chinese ecosystem. So again, many, many reasons why the traditional way of growing food protein is becoming unsustainable. So of course, this is where cultivated meat comes into play, also known as in vitro meat or clean meat or cellular agriculture. There's many different monikers for this type of technology, where this approach is thought to address a variety of these unsustainable resource usage issues. And basically, cultivated meat involves taking a sample of cells from the host animal, a food animal such as cow, pig, or fish, growing those in the lab in certain conditions that allow those cells to sustain themselves, and then also adopt an architecture that is compatible with something that would be edible. So this requires a lot of tissue engineering and optimization of how to maintain those cell growth and also the cell architecture. So this is basically the technology behind cultivated meats. And this estimate was done, it's published online. You can find it by a firm called CB Insights. Again, this might be accurate or not. This is just an estimate that was done many years back that lab-grown meat or cultivated meat could dramatically reduce water use, dramatically reduce greenhouse gas emissions, land use. But then on the flip side, it's more expensive to produce a given pound of meat in this analysis compared to traditional agricultural sources of that pound of meat. So obviously that's the issue with hindering this industry, but it is changing over time. Various firms and academics are addressing this, and we hope to have done a little bit of our part to address this cost disparity. So it's known that there was a survey done by GFI recently that firms in this space or companies in this space attribute about 50% of their costs to the cost of media to grow cells. So what is media? It's a broth, basically, that your cells need to grow in that contains components necessary to sustain their life. And within that media, traditionally, an important component of that media was fetal bovine serum. So as the name implies, this is derived from young cows, their blood, and it contains a variety of components that cells need to grow: hormones, vitamins, minerals, lipids, and also what are called growth factors. So I'll talk, obviously this is the subject of my talk, and I'll talk much more about those. But at this point, let's understand that these are important components in media to sustain the cells, and FBS traditionally has been a source of those growth factors. Of course, there's issues with this, right? If we're trying to get away from using animals to produce food protein, we want to remove FBS. So that's kind of contradictory there, right? So we need an alternative source of the components that FBS is contributing. As well, it's costly. I checked a couple of days ago, it's about $1,000 in Canadian dollars to get a liter of FBS. So that could be cost prohibitive if you're scaling up large bioreactor scale of cell culture. As well, it's known that there's batch to batch variation. So any given batch of FBS might not perform the same in terms of maintaining the growth of your cells as a previous batch, for example. So there's a variety of reasons why we need to get away from using FBS as a source of these essential components to grow cells. So there are solutions for that. There are defined serum-free media, defined as in the components are clear and produced chemically or recombinantly from alternative sources. So there's a variety of these that are available and they're on the market, but these can also be decently expensive. So I just checked a few days ago, about as much as $600 for some of these medias per liter, Canadian dollars. So again, serum-free medias, they exist, but they still need work in terms of reducing their cost and also the applications that they're used for. So some firms have completely eliminated FBS from the media, such as Upside Foods and Mosa Meat. Some of this technology is published, some is not. Mosa Meat recently published a lot of their research in Nature Foods recently, but other firms, it's not really clear how they're achieving this. So from an academic perspective, there's still some issues here. And also from the growth factor perspective, we wanted to see if we can play some role in reducing the cost of the growth factors in media. So an analysis was done a couple of years back, but I would say it's still fairly relevant today. It's only a couple of years, by Liz Becht from GFI. And you can find this online also, where she did an analysis of the components in cell media and what are the drivers of cost of that cell culture media. So this is a plot I made from her numbers in that study. And this is the different components of Essential 8 defined serum-free media. And in the blue bars, you can see that those are the liters, and her analysis was done on a 20,000 liter scale. You can see that in terms of liters, it's just the volume of it. It's mostly from the basal media and sodium bicarbonate. But on the cost perspective, on the orange bars, you can see essentially the basal medium and sodium bicarbonate are cheap. They're not driving the cost. But then on the flip side, there's those two components at the bottom, the growth factors, FGF-2 and TGF-beta. They actually comprise more than 99% of this particular media, but they're actually much less than 1% of the volume. So obviously, these are a major cost driver of this particular media and other medias, which also need to have growth factors in them. So that kind of was the impetus for us to kind of get into this field. So what are growth factors that we've mentioned quite a few times? So growth factors are small proteins that all eukaryotic or multicellular life forms need to grow. So they're used for a variety of purposes, for cell-to-cell communication, for creating a particular architecture in a tissue where cells signal to each other to undertake certain behaviors. And these are essential for the growth of eukaryotic cells, any cell that you would use for cultivated meat. So growth factors are encoded in the genomes of these species, and they're expressed, the proteins are expressed and secreted outside of the cell, but then those growth factors will come and bind to, let's say, a neighboring cell and bind to their particular receptors. So just shown in this graphic, there's little green circles, the growth factors, binding to the receptor that's on the membrane of the recipient cell. And then this initiates a cascade within the cell called a signaling cascade that ultimately results in turning on or off particular genes that then result in a particular functionality that the cell will take. So either this will signal the cell to proliferate, to divide. Growth factors can signal for differentiation, for them to change their architecture, change from, let's say, a stem cell into a different type of cell, a satellite cell or a myoblast, et cetera, along the pathway of maturation of tissues. And growth factors can also signal for apoptosis, which is a form of controlled cell death. And growth factors, there's a lot of different families of growth factors. I'm just showing 3 of them here. And in our analysis, we studied 10 families, but there are more. So just some schematics of the structures of these growth factors. There's PDGF, FGF1 and 2, and TGF-beta, or transforming growth factor beta. So I mentioned, according to Liz Becht's analysis, also that growth factors are really the cost drivers within serum-free media. And why is that? Well, in commercial media, if you were to buy off the shelf serum-free media, it will have growth factors, but those growth factors were recombinantly produced. That is, some microbe or cell line was engineered to produce this growth factor, and then the growth factor was purified out of those cells. Sometimes that is expensive for particular growth factors, and I'll talk about that through the talk, particularly TGF-beta. Recombinant production of TGF-beta is very expensive to buy off the shelf as well. And I notice kind of the growth factors that you buy, they're always human, mouse, bovine. They're restricted in the species type. And from a biochemistry perspective and structural biology perspective, which is my background, if we're really interested in studying a particular protein, but we know that protein to produce it is expensive or difficult, we often switch to studying a homologue from a different species, a related species. For example, if you're interested in a human protein, often people study the mouse ortholog or a rat, or you even go to a yeast protein in some cases if they're similar enough. So from my perspective, when I was kind of writing the grant and getting into this field, I thought, well, maybe there's growth factors from other species, like cow, for example, that we do use for cultivated meat. Maybe we should be focusing on cow growth factors or horse or others, or even fish. And I thought maybe there's something there. Maybe we can produce those cheaper. That was the first point. And then the second point is sort of related, is some of these growth factors are difficult to produce recombinantly in what we consider the fastest and cheapest, easiest protein expression system. And that's E. coli bacteria. This really is the workhorse for protein expression if you can purify your protein in E. coli. Again, not everything can be purified from E. coli. So these growth factors are obviously eukaryotic genes. Sometimes E. coli has trouble with that. Most of these growth factors have disulfide bonds. So these are links internal inside the protein sequence, and they form a bond in the three-dimensional structure that is necessary to create the three-dimensional structure of the growth factor. Sometimes E. coli has difficulty with that, figuring out what are those proper linkages. And if it gets it wrong, the protein misfolds and you lose it. And then some growth factors also have other modifications that E. coli just has difficulty with, like glycosylation is a big one. E. coli does not. Glycosylation is adding a sugar chain onto your protein. E. coli just doesn't do that. And lastly, some growth factors are known to have some inherent stability. So either proteolysis, they degrade, temperature, or certain temperatures, they may lose their functionality, or pH, and that may have issues for cultivated meat applications. So to our grants, it was awarded in 2018, 2019, and we had three main goals. The first goal was to identify growth factors that can be inexpensively purified in E. coli. And we leveraged our years of experience in doing that for many types of proteins. We thought that we could find, again, this idea of finding orthologs or homologs of the growth factors in other species. We wanted to try that in E. coli. And then secondly, we wanted to take that set of growth factors and ensure they actually function to maintain the growth of cells that are relevant to this industry. It's not really much use if you can produce a growth factor, but it doesn't work. It's non-functional, right? So we wanted to make sure that these these growth factors from different species would actually work to sustain cells of relevance. And the third goal, which actually, for the lack of time and resources, we didn't get to do much for this goal, but we are continuing this research, is we wanted to actually change the sequence, the DNA sequence coding for these growth factors to engineer synthetic or non-natural growth factors. From a bioengineering perspective, there's no reason to restrict ourselves to the native sequence, the wild type, so-called wild type sequence of these growth factors. What if we can change that DNA sequence and create some non-natural growth factor that may have different properties? And from a genetically modified organism perspective, I think this would not be a major hurdle because growth factors are not internalized into cells. They're on the outside of cells in your media itself. Basically, you can wash them away. So any non-natural growth factors that are created don't get integrated into the cells that you would or the tissue that you would consume. But like I said, we haven't been able to do much in this, although we do have this as a major research focus going forward. So yeah, ultimately then we want to create new growth factors for use in any type of cell culture application, in particular for cultivated meat. So our research had basically three phases. The first phase was production of the growth factors. The second phase was to evaluate their bioactivity. And the third phase, which again, we didn't really get to, but we hope to, is to engineer those sequences, to change the sequences themselves, such as creating hybrids. So on the phase one side, the first step that we did was we undertook some in silico or bioinformatics work to look at the families of these growth factors and just kind of sample the diversity of these families across different species. A lot of species genomes have been sequenced, and this is, big thanks to all the public sector funding that has been devoted, such as from NIH, a National Institute of Health in the US, for sequencing genomes that we could then use for purposes like this. Then we got those genes synthesized by a company and cloned into an expression vector to test for expression in E. coli on a small scale. Then we purified any of the candidates that had good expression solubility characteristics. And then finally, we fed those into the phase two, which was evaluating their activity, sustain the growth of 3 different cell lines. And I'll talk about each of these steps. So just a couple snapshots of the growth factors that we targeted, primarily because of their role in the growth of muscle tissue and also their price for recombinant, just buying them from a commercial vendor. These are quite expensive. So first that we targeted was insulin-like growth factor 1. This is a 70 amino acid protein, so it's very small. Its 3-dimensional structure is shown on the right. And it has 3 of these internal disulfide bonds. So again, E. coli sometimes has difficulty creating these bonds to stabilize the structure of the protein. It is present in FBS and in serum-free medias. So it binds to its dedicated receptor, the IGF-1 receptor, and also to the insulin receptor to initiate signaling that ultimately leads to cell growth. And just a quick survey I did recently, it's approximately $800 to buy a commercial, 800 Canadian dollars to buy per milligram. We also targeted transforming growth factor beta 1. And this is a 112 amino acid protein. It actually has 4 disulfide bonds, one of which is across the 2 chains of the protein. I show the structure on the left. And again, for a small protein, that is a lot of disulfide bonds, and E. coli sometimes has difficulty creating those, leading to misfolding of the protein. And it's found in FBS and serum-free media, and it supports undifferentiated cells. It has really complex biology. I'm not a TGF-beta expert. It has, and there's also a superfamily. There's beta 1, beta 2, beta 3, etc. There is very complex biology, but anyway, TGF-beta is an important growth factor in serum-free media. It binds to its receptor and activates a signaling pathway in cells. And this one's super expensive to buy. It's $7,000 Canadian dollars per milligram. So the bioinformatics selection, so this is just a snapshot of a tree using the protein sequence of PDGF from a variety of species. And we did a similar analysis for 10 different families of growth factors shown on the bottom right. And this is just basically a visual way to show that the sequence of these particular growth factor varies according to the sequence that you're talking about. So mammals cluster at the top. There's also, bat, hedgehog, rat, etc. Fish are nearby, but they do have this distinct grouping. So we targeted a variety of PDGFs from fish, also from some reptiles or amphibians, and also from birds. We were really non-biased in terms of the species that we targeted. We just wanted to sample the diversity of a sequence family, thinking that, well, if it's very expensive to produce human growth factor or bovine growth factor, perhaps a duck would be cheaper. And whatever species that came through our analysis, we would then go forward with that. So we have a variety of different species. And again, we were really agnostic for what came up. And I should also mention that the sequence diversity across this phylogenetic tree and the other ones that we did spans about 50 to 90% sequence identity. So there is some diversity in the sequence here. And from our experience, that could mean changes or different properties for purification as a recombinant protein, and also different functional properties. So that was good from our perspective, that there was a significant sequence diversity here to sample. So then we, through the bioinformatic analysis, we targeted 152 growth factor genes. These were purchased as synthetic genes from the company Twist Bioscience, and these were codon optimized for expression in E. coli. So this is basically the DNA sequence was altered, not to change the protein that would result, but just to change the particular tRNA or codon that was used in E. coli to more suit what E. coli is usually used to. And we thought, and for some cases, that really helps for protein expression and solubility. So those were cloned into expression plasmids, and some E. coli strains were transformed, and then small-scale expression testing was done. So this is a schematic, and once again, all of this is in our bioRxiv paper, and it will be in the peer review paper when it comes out. This is just a schematic of some of the expression constructs that we created for these growth factors. And I just want to bring your attention to, let me pull out my pointer. I should have done that sooner, actually. Isn't there a annotate? I don't know if you guys can see this. Oh, I don't know if you can see my mouse.
Yeah, we can see your mouse.
See the mouse? Okay, excellent. I'm not super used to the that function in Zoom. Anyway, so I wanted to bring your attention that we target different fusion partners. So often for protein expression, you want to have another protein that is known to be able to fold in E. coli to help your targeted protein also fold and be soluble in E. coli. So that's why we used a variety of fusion partners. And I wanted to bring your attention to DSB-A, DSB-C. And this was a major innovation for us. And this definitely I give a lot of credit to Cam Semper, a postdoctoral fellow in our group, who came up with this idea. So DSB-A or C is a bacterial protein that's a disulfide bond isomerase. And that actually turned out to be very important to help form those disulfide bonds, particularly in IGF and TGF-beta. And we also had histag to enable affinity purification. And then the host strains that we used, I'm showing a couple here, BL21 Gold, which is our workhorse for protein expression. And we also use Shuffle T7, which is a strain designed by NEB Biolabs. And this actually has the DSB, I think A, or I forget which DSB it is. That gene is integrated into the chromosome of E. coli. So it's thought to also help E. coli produce the proper disulfide bonds for eukaryotic proteins. So we use Shuffle T7 as well. And then basically we did this screening for solubility, and we achieved pretty good results for soluble growth factors. And in particular, highlighted in the red stars, we achieved 9 soluble IGF 1 or 2 growth factors, 7 TGF-beta1s, and 2 myostatin and 4 LAF, which are TGF-beta superfamily growth factors also. And those are also important for muscle cell biology. Oh, I have this annotation thing going on here. What's going on? And I can't find my mouse. I turned this annotation thing on, and now I'm kind of, okay, here we go. So, and again, this is from our paper also, but this is just an example showing successful soluble expression and purification of DSBC tagged IGF 1 and 2, shown on the left. And on the right is DSBC tagged TGF-beta1. And we're just showing a subset of growth factors here from different species. And basically, they achieved soluble expression at the expected molecular weight, showing that indeed we could produce these. And it really was this DSBC or DSB-A tag. They were interchangeable. These fusion tags were really instrumental in helping these proteins fold properly in E. coli. And we also used Shuffle T7 cells for this. So then with our approximately a set of 50 growth factors, we then proceeded to that second phase, which was the bioactivity testing. So we grew NAH3T3 fibroblast cells in the lab. These are mouse cells. And actually a reviewer for our article, it's under peer review, said, well, these are not super relevant cells. We knew that from the beginning, but they're really easy to grow and super cheap. And as a platform for screening a large number of growth factors, we started with that. But then recent collaboration and recent results have actually shown that our growth factors work on bovine satellite cells. And I'll show that data in a few slides. And also Cam and our group at Calgary has been working with our recombinant growth factors using fish cells. So these are CHSEF embryonic derived cells from bluegill. And indeed, at least for FGF2, our growth factors are functional against those cells. And then our assays for bioactivity, we're measuring proliferation by a respiration assay, and also immunoblotting to look for activation of those signaling pathways, which I mentioned. These growth factors will turn on certain signaling pathways. So we did Western blotting to make sure that those pathways are indeed activated, as we would expect. So this is a lot of data. The main sum, the bottom line is that our purified growth factors from a variety of different species with these DSBC tags are actually functional against 3T3 cells in this case. And again, there's a lot of data here. This is in our bioarchive paper and it will come out in the peer review process. But we did find that actually some candidates have interesting properties where they seem to have higher potency than the commercial growth factors that we bought. So I'm highlighting those in red, where it seems like particular FGF2s that we made, for example, from salmon, had higher potency in terms of, and I should mention, sorry, that the y-axis is fold change in proliferation relative to the baseline. And yeah, so for example, salmon FGF2 at low concentrations has higher effects on growth than the commercial equivalent. We found some PDGF-BBs also that had good characteristics, IGF-1, etc. So the sum to us was that this proved to us that the growth factors work, and also some are more potent at lower concentrations. Excuse me. And I should say that this experiment was done by serum starving these cells to a very low percentage of FBS for 24 hours. And then the growth factors were added for 48 hours before an MTT, which is a respiration assay, was done, a colorimetric assay was done. And also shown in the TGF-beta1 graph, the DSBC tag, I mentioned that there's this tag on the growth factor. That protein itself, we confirmed it does not activate growth of cells, but in the presence of TGF-beta with this fusion partners, DSBC, it works. And actually you can cleave off the DSBC and just have the free TGF-beta1s, and those also work. But for our perspective, the tag can stay on. That alleviates additional purification steps because it allows proliferation of these cells. And then I mentioned that we wanted to make sure that the signaling pathways were functional using these recombinant growth factors. And indeed they are. So on the left, I'm showing the a very simplified signaling pathway that's initiated by the FGF2 growth factor, IGF-1 and 2, and PDGF-BB. Those activate phosphorylation of a protein called ERK1 or ERK2. So if you look for phosphorylation of ERK1 and ERK2, you know that it's been activated. So in the middle, 3 Western blots in the middle, those show that those bands do show activation of or phosphorylation of P44 subunit of ERK and the P42 version of ERK, whereas the negative control, the non-growth factor treated cells, do not activate the ERK, phosphorylation of ERK. And that was true for FGF2, IGF-1, IGF-2, which had the DSBC tag, and PDGF-BB. So that was good, showed that these growth factors are indeed turning on the signaling as they should. And then finally on the right, I'm showing that TGF-beta1 with the DSBC tag. And in this case, it activates phosphorylation of a protein called SMAD2. And if it's active, if it's binding to the TGF-beta1 receptor, and the Western blot on the bottom is indeed showing that phosphorylation of SMAD2 is detected, whereas the negative control, untreated cells, do not activate phosphorylation of SMAD2. So this was great. So this proves again that our growth factors do the job that they're supposed to, at least in this assay, and with these simple cells, they turn on the activation of these intracellular proteins. So during the peer review process, our paper was in bioRxiv and we submitted it to a journal. And as I mentioned, a reviewer, questioned our use of 3T3 cells, but we were aware of that. And we were contacted by a group in Aarhus University. And I'm showing a picture of Stig Skigergaard, who actually is a PhD student who was interested in using our growth factors and testing them, their effects on bovine satellite cells. So these are primary cells derived from cow, and members, I'm sure Stig will be able to answer any questions about that. I'm not sure if he's here, but anyway, if you have any questions about, the extraction protocol, et cetera, I didn't do that work, but Stig did that work in Denmark. But he contributed some amazing data, which we're adding to our paper to fulfill the peer review process. Where actually our growth factors also work to turn on metabolism and respiration on these cells. So again, he took these bovine satellite cells, serum starved them to 0% FBS, and then added our recombinant growth factors for 48 hours, and then did a respiration assay. In this case, it was WST1. It's also a colorimetric assay. If the cells are respirating oxygen, your mixture will turn a different color, and you can read that. So relative to the control, you can see on the right that he has a control where increasing amounts of FBS turns on the metabolic activity, the respiration of these cells. But so does our growth factors. So actually, many of our growth factors turn on metabolism much more than 0% FBS would, which basically told us that these can replace FBS. And again, this is just in a very simplistic system with only this growth factor added. And this really opens up doors now. And I'll talk about this later. Can we combine these growth factors? What are these growth factors doing to the cell, morphology, et cetera? But this was great data to us. And again, it shows that, growth factors, some of which have this DSBC tag, and they're derived from different species, they work on bovine cells. And that's great. And then finally, in terms of the bioactivity testing, and this was done by Cam at our group site in University of Calgary, who's a postdoctoral fellow there. And he's been working on trying to derive a serum-free media for use in fish cellular agriculture. And he used recombinant FGF produced according to our protocols. And he tested again for activation of this ERK signaling pathway. And he found, again, that a variety of our recombinant growth factors, he was targeting fish growth factors, they also activate the signaling pathway. And in some situations, higher activation of the pathway than, for example, human FGF that you would buy from a company. So yeah, again, this is work in progress, and we're testing more of our growth factors against this fish cell line, but it also is promising that, hey, our recombinant growth factors, some of which have the tag, may have potential to allow for proliferation of fish cells. And the final thing that I want to talk about is what does this do for the cost of media and producing these growth factors? So now that we've established protocols for recombinant production of these growth factors, and we've established that they have promising bioactivity, what does this do for cost? So if you just look on the recombinant protein production basis, what is the cost for this? And for different growth factors. So I just we did a little cost analysis of how much does it cost us to produce a milligram of our growth factors with our protocols relative to what you would buy off the shelf. So again, I understand that this is, we don't have any profit margins here. This is on a lab scale also. There's a lot of other issues. We have, we're not proposing that we would sell at this price. And also there would be validation in terms of, inspections, et cetera, quality control that we are not doing in our lab scale. But still, I think that there's something very promising here that, especially for TGF-beta1, we can produce for $10 a milligram. But as I mentioned, that if to buy it from these companies mentioned here is in the thousands of dollars. So that is a huge cost difference. Again, with the caveat that the quality control, there's additional quality control steps that would be necessary for commercial grade or food grade, certainly. Of course, that would increase the price. But to us, this was extremely promising. And the same for IGF-1, PDGF, and FGF. And then if you take those cost of production of growth factors, if you go back to that analysis that I presented at the beginning of the talk, done by Liz Specht from GFI, can we plug in our numbers for recombinant protein production of growth factors into the essential 8 media mix? If you remember, FGF2 and TGF-beta were 99% of the cost. Well, with us, it lowers down to about 8% of the cost. And the total cost then of the media is about 26-fold less. Again, this is it's kind of a crude analysis. There's other steps involved for, if you're producing growth factor en masse, there's other factors that you would need to include there. But still, we think this is promising for not only lowering the cost of production of the growth factor itself, but also lowering the cost of a minimal serum-free defined media, such as Essential 8. And that brings me to my conclusion. This is everything that we managed to accomplish in our research. So we've shown that all major growth factors that we targeted can be inexpensively produced in E. coli. And the major innovation for us was the DSBC tag and the use of the Shuffle T7 E. coli cells, which really help E. coli produce, in particular, IGF-1 and 2 and TGF-beta1. We also discovered this phenomenon of cross-species activity, which was really never explored. We showed that fish, mammals, bird growth factor, all of our growth factors were active against the 3 cell types that we use. And this opens up further lines of research. And also another thing for the engineering point of view, what is it about the sequence diversity here? Some species have a little bit different sequence of growth factor that enables it to be, different properties. So that's another phase of our research. But it was a really interesting result that there's this cross-species activity. As well, we showed that at least with the cell types that we used using our assays, that our growth factors are functional against these cell types, including with the DSBC tag, which was pretty key for us. And then according to our techno-economic analysis, which is simplistic, but is promising, there is this at least 20-fold reduction in production cost of growth factors, and actually more than 700 times for TGF-beta1. And according to our calculations, we think that this would drop the cost of serum-free media by about 26 times. And that's pretty significant. So yeah, this really is kind of the beginning of our research. There's a lot more to do now with this toolkit of growth factors. We're interested in continuing to screen them versus more cell types. We'd be happy to share these growth factors, the expression constructs, and the protein itself with you if you have interesting cell types you'd like to evaluate them on. Also, we didn't really focus on analyzing the morphology of the cells, the differentiation. What are the cells doing in response to the growth factors? Is that something that is compatible with further cultivated meat and tissue engineering? Is there any utility there? So again, this is opens up the doors now for that work. As well, we have this idea of changing the timing of adding these growth factors in cells and maybe mixing them and combining them. This may better mimic a natural environment where you don't necessarily have just all growth factors present at any given time in the in the mix of any given cell. No, biology has a synchronicity and a purpose for secreting and activating certain growth factors according to given cues. So with these tools, we'd be interested in kind of doing a matrix of combining these growth factors. And then finally, as I mentioned, we were hoping to get to, and hopefully we will, is we'd like to then manipulate the sequences of these growth factors using some of the insights from this research to create new non-natural growth factors. And there's various ways that you can do this, such as directed evolution or machine learning. And maybe we'll create some non-native growth factors that may have even better performance in terms of recombinant protein production and maybe bioactivity. Yeah, and finally, I just want to mention that this was a completely open science effort, and Good Food Institute in the research grant program we were a part of, that was an essential component of it, where everything is communicated openly, all our reagents, all our techniques, and the knowledge is open to anybody in the private or public sector. So again, if you'd like any expression constructs or purified protein or protocols, feel free to contact me or Cam Semper. The email addresses are there, or you can contact me later. And yeah, we'd love to share. And this, for us, we're not tissue engineers, so anybody out there who would like to use our growth factors for further applications, we would love to get in touch with you. Yeah, that's it. And so this was a major team effort. So I just want to thank and acknowledge everybody that was involved. Mina was Vikatasan was a research associate in our lab who really was spearheading the protein production, screening, Western blotting in our sites. Also Allison McGuigan, who's in BioZone, helped with tissue culturing and use of her facility. Natalie and Rosa were really instrumental in the cloning and screening of growth factor expression. Cam Semper's postdoctoral fellow, who really was instrumental with the innovation of the DSBC tag and also screening against fish cells. And he also has been funded by New Harvest. And Stieg in the group of Martin, Jette, and Margaret in Aarhus University, kind of latecomers to our analysis, but really instrumental in providing some super relevant data for the utility of these growth factors against bovine satellite cells. So thank you very much to you in Denmark as well. And finally, to everybody at GFI that has helped us over the years, Aaron, Tom, Elliot, and Liz, really instrumental in helping us with the grant program and technical assistance and making connections in the community. So thanks very much. And that's everything for me. Yeah, and I'd be happy to take any questions that you like.
Awesome. Wow. Thank you so much, Peter. This was incredible. And just, yes, team open science all the way. We can stop sharing your screen now, and actually would love if you could maybe turn your video off and restart it. I think you froze a few minutes back, and that might reset things a little bit. You sounded perfect the whole way through, but there you are, back in motion. Okay. What an incredible set of research findings. It is incredible to know that we were able to kind of, the very royal we in this case, able to kind of produce all major growth factors inexpensively in E. coli. Incredible that there was this kind of cross-species functionality, and remarkable that there was a 20 to 26 times cost reduction. And thank you so much for your orientation towards open access science and willingness to kind of share all of these protocols and growth factors and everything you've learned with the rest of our community. It goes such a long way to accelerate the pace of technological progress in this field. Lots of enthusiasm throughout the chat, and also many questions in the Q&A. So we will move to Q&A now. Just a reminder to audience members, if you haven't yet already done so, feel free to add any questions you have for Dr. Stogios in the Q&A section. There's also an upvoting feature whereby you can vote on the questions you're most interested in having answered. We will wrap up Q&A at the 15-minute mark, 15 past the hour. And at that point, any folks who are interested in joining us for a networking session with scientists across the globe are welcome to do so. And Renee will drop a note in the chat there. I also forgot to mention this early on, but in case you haven't yet seen it, GFI did recently release our 2022 RFP. Phase 1 applications are due June 3rd, which is really right around the corner. So Renee will drop a link in the chat to that RFP, and we encourage everyone to take a close look at that and circulate that around your networks. Alrighty, with that said, Peter, so many questions here in the Q&A for you. One question is around regulation. So to the extent that you feel able to speak to this, do you know if there are more regulatory hurdles if you alter the amino acid sequence of the growth factors in question?
Yeah, this is not really my expertise. And yeah, some of the other questions also I've just been scanning, so I don't want to say the wrong thing. So from my molecular biology and cellular biology experience and education, I don't think there would be, just because growth factors are not internalized into cells. So you should be able to wash them away after they've done their job of activating proliferation of differentiation. But again, that's even that would need to be tested, right? You'd have to evaluate where do the growth factors go, how much of it can be recycled in the media, etc. So assuming you can clean them up away from the cells, it should not create any regulatory hurdles. But again, like that's to the best of my ability to answer that question.
Thank you, Peter. Yeah, you have a royal pass on anything that I ask you that you feel like is outside of your wheelhouse. But thank you for answering that question from your perspective. There's a question here from Thomas. How did you solve the issues with respect to correct folding of E. coli and those disulfide bonds? Did you do it post-harvesting with the recombinant proteins in vitro?
No, E. coli figured it out for us. We didn't have to do anything. It was that DSBC tag really helped, and then the shuffle T7 cells, which express the DSBC protein. That's the job of those isomerases, right? They figure out the proper way, and the mechanism. Any misfolded protein would go into inclusion bodies, and we wouldn't be purifying those. We're always cracking open the cells, and anything that's soluble is what we purify out. So yeah, the DSBC system internal to E. coli, it figured it out for us. So we didn't do an estimate. Was it 100% effective in recovering all of TGF-beta, for example, to be soluble or not? We didn't actually do that, efficiency calculation. So it actually may not be super efficient. It might be, on the order of 50%. We didn't do that. But suffice to say, it did solve the problem, and it created soluble protein that we could extract once we, lyse the cells.
Awesome, thank you. Question here from Eric. Is there a library or database of annotated protein chaperones that aid effectively in folding and refolding?
That's a really good question. There's certainly literature about this, and some vendors, some companies do have information about recombinant protein production, and that will include information about chaperones. So if you do some Googling for recombinant protein, like fusion tags, you should be able to find, for example, I think Thermo Fisher would have some, NEB would have some. So some of the commercial companies would have some information, and certainly literature. But yeah, that's a really good question. It'd be good to have just a database to say, here's all of these chaperones. I imagine if you look for reviews in the literature, that would be a good way to go.
Thanks, Peter. All right, there's a question here about the bioactivity assay. How was the relative fold proliferation rate calculated? Were you normalizing the no growth factor background growth level?
Yeah, depending on the cell types that were used, and you can find those details in our paper. For example, I think for 3T3 cells, it was normalized to no growth factor added. But yeah, that technical information is in the paper. So we have for the different cells in different labs, we had different normalization standards. For example, you can also make it relative to 0% FBS. So yeah, it depends on the cell, but that you can find that information in the paper.
Awesome, thank you. Were all of the growth factors expressed and purified under the same environmental conditions?
Yeah, so different E. coli cells, certainly. For example, these Sheffield T7 or BL23 Gold's E. coli cells. So there are different strains of E. coli. But other than that, the same, lab environment, similar temperatures. We had to undergo different rounds of affinity chromatography purification for different growth factors. For example, PDGF needed to go through a second column to remove, in that case, it was a thyrodoxine fusion tag. So the cells themselves, it was E. coli cells kind of grown in the same environment. And then for some growth factors, there were some different chromatographies that needed to be done. And all that technical detail is also in the paper.
Great, thank you. A question here from Ellen Mosho. You stated earlier that the bovine satellite cells were first serum starved prior to the introduction of growth factors under investigation. How did you make sure the cells didn't differentiate during this process? Or did you kind of wean the serum off stepwise with increasing amounts of growth factors?
Yeah, this would be a question to our team in Denmark. I wish I could answer that. I didn't do that experiment, but so it's not in our bioarchive, but we're trying to get it into our peer review paper. So then you'll find out more about that. We anticipate it to go through the peer review process relatively smoothly. So that will be in there soon. So yeah, sorry, I didn't do that experiment. And off the top of my head, I can't answer that, but it will be published and it will be made available.
Awesome. Yeah, no worries. You are not expected to be the authority on all things. There's a question here from Yete Young from Aarhus. Great results, Peter. Do you want to pursue research using other vehicles than E. coli, or is this just the best study species or specimen for you to use?
Yeah, hi, Yetza. Thanks for that question. Yeah, that's a great question also. From just the makeup of our lab, and typically what we do is E. coli. We're really specialists in the different E. coli strains and vectors, et cetera. But certainly there's other species out there. For example, Pichia pastoris is a yeast that is used for protein expression. Certainly all of these could be tried in that species. I mean, we don't have the funding for that or the manpower currently for that. But certainly it'd be interesting to see, yeast or CHO cells or HEC cells, et cetera. There's many different recombinant protein expression cell systems. Yeah, I'd be interested in seeing how they perform. But yeah, like just for the sake of resources and just our lab currently as it's constituted, we won't be doing that. But yeah, anybody else would, like I said, all our constructs will be available, and certainly you can plug and play those growth factor genes into your own favorite expression system. So it'd be really interesting if people were to do that.
Thanks, Peter. There's a question here about Matrigel, which I am not intimately familiar with, but many people developing serum-free media seem to use Matrigel to grow their cells. Matrigel is loaded with growth factors and many other growth-promoting factors, and this person is asking what your opinion is on this.
Yeah, I'm not familiar with that either. I imagine that there'd be space to change the growth factors in the Matrigel, for example. Somehow those growth factors have to be produced and integrated into the gel. So you may still have some issues with the recombinant protein production cost there, in which case our growth factors may be of value there, and you might want to change the species involved. So yeah, that's a really cool idea. And I mean, I'm not familiar with it, and certainly I'm not going to be doing that, but certainly, yeah, as we hope to have these growth factors fertilized in the field, it would be great if somebody uses our protocols and tries using our growth factors with those.
Do you have, I don't remember seeing any data on the yield of growth factors per kind of liter of culture. Do you have any data on that?
Yeah, it's about, on average, it really depends on the growth factor. It's about 5 milligrams per liter of E. coli. And then for the bioactivity purposes, that's plenty. I mean, you use nanogram amounts for those assays. So, in terms of somebody who's a biochemist and structural biologist, 4 to 5 milligrams per liter of E. coli is okay. I mean, it's not spectacular. We've been able to purify proteins on the hundreds of milligrams per liter scale, but for this purpose, it's sufficient. And even that could be an avenue of future optimization, right? So optimizing the growth of those cells in a bioreactor environment or fed batch system, certainly those yields could be improved. But from what we did, we could achieve about 5 milligrams per liter on average across these growth factors, some better than others.
Mm-hmm. There's a question here around whether you have any calls to action for maybe policymakers or other kinds of ecosystem builders in our space. What do you think other actors, maybe especially on the regulatory side, might be able to do to help any of these efforts along? And again, totally appreciate if this is just not something you want to comment on deeply, but some examples they kind of raise here are, reducing or subsidizing costs, increasing bioreactor infrastructure, promoting and funding more open access research, et cetera. Just this attendee is curious to get your perspective on what the most impactful thing a policymaker might be able to do to advance your research?
Yeah, so it could be a number of things. It's hard to say what would be the most. Obviously, it's, you have to try and see what happens with some intervention from a policy perspective. But from a science perspective, funding for this type of research is very important. Right? We couldn't do any of this work without the GFI and New Harvest and some other funding agencies. So, and even there's not that many funding sources for just, pure science and open science on top of that. It's hard to find funding for that. That is changing. I know in Canada, our federal funding agency called Genome Canada is funding cellular agriculture research, and that's huge, right? So, and they're investing significant amounts of money into that, right, for academic or industrial research also. So, definitely more funding for this research. And then on the open access side, that's also very important. Open science side, yeah, that's also a big deal. For us in academia, it's the IP, dealing with IP issues slows us down tremendously. Going through a bureaucracy in our system, in our individual institutions, really slows things down. And I can understand there's a purpose for it, there's a role for it, but if you're talking about making innovations, and you're waiting for funding to come in, and you're waiting for MTAs to go through months and months, like there's hurdles. So breaking the kind of red tape and helping, at least on the academic side, helping us accelerate dealing with those issues. So that's where open science really is a radical way of looking at things. It just says the hell with it. Like, we don't care about that stuff, right? Especially at the level of pre-commercialization. Let's just put it out there, and the whole environment, the whole ecosystem can use it. And if you think about it, where have our revolutions come from? In the years, like mRNA vaccines, that was publicly funded research that was, public, that was published and open for anybody. And nobody ever thought, 10, 20, 30 years ago, when that research was published, that this would result in a vaccine that would stop a pandemic, basically, or hopefully stop a pandemic. We're still not done, and you can say the same about CRISPR-Cas9, right? That was, microbiologists interested in how bacterial immune systems work, just from a pure, curiosity and basic science perspective. And now look what it's done for genetic engineering of all kinds of systems, right? And again, that was funded by public research dollars, and that was advertised, that was published. Of course, there was a lot of legal battles and about patenting, etc. I won't get there, but still, what I'm trying to say is public research dollars to then allow research to be communicated is transformative, and it has been transformative. So we need that in our space, and it's coming. Like I said, in Canada, the government's getting on board. So yeah, and then open science movement is definitely growing as well. So yeah.
Yeah, thank you so much.
Some of your question there.
Yeah, no, that was such a powerful set of reflections. And of course, we feel very deep resonance here with your rally cry around open access science, and also for the great need for more public research funding. And I agree that I think there's a tractable path forward here with some pretty historic funding milestones coming out of the Netherlands and other governments, hopefully also rising to the occasion. Awesome. We have just a few questions left here. And I do want to invite you, Peter, to take a look through and see if there are any that you're most excited to answer. And there's a question up top around the techno-economic analysis and curiosity around whether it was based on bioreactor or flask production of the growth factors.
Yeah, so this was flask production. So this is what we did in our lab. So this was, 6-liter flasks of E. coli. So certainly, like I mentioned, kind of, caveating there during the talk, there is additional optimization that could be done, right, in bioreactor scale and, different, situations there. So our techno-economic analysis was with our setup, but yeah, that's not applicable to all environments. And then, yeah, I saw your question also asks about licensing fees. Yeah, I never actually thought of that. Yeah, that may be an issue since those are commercial cells. So, I mean, Maybe, yeah. So in our analysis, we're not going to change it to integrate that because, we're not selling these growth factors, right? But certainly if any of them were to be commercialized, yeah, things like licensing fees would have to be integrated for sure.
Mm-hmm. Awesome, thank you. There's also a question here around, let's see, lots more regulatory stuff. I think you answered this question around biggest contributors to the current cost of growth factors production quite a bit in your talk, but is there anything you want to illuminate further around the kind of bottlenecks that still remain when it comes to bringing these cost drivers down?
Well, from our perspective, like I mentioned, some growth factors are very expensive to buy commercially because they have to go, the companies have to go through, they have to use eukaryotic cell lines, or they have to use multiple purification steps, or the yield is low, or the proteins misfold for a variety of reasons. So we think that achieving pretty good success in making soluble, let's say TGF-beta1 and IGF-1 and 2 should get around that. Of course, with the caveats that, this is our lab scale basis. There needs to be, verifying that is scalable to commercial quantities, et cetera. So yeah, just like the technology for, and I should say the success rate in producing protein. Yeah, so we hope that what we've done has really, solved that problem, at least for specific growth factors. And then, we didn't do a lot of work on this, but some growth factors have, like I said, there's some issues about stability and proteolysis. So yeah, let's say you'd be able to purify, this is for FGF in particular, purifying FGF, but then it might not survive during the length of your cell culture experiment. So there's further work that could be done there. There has been work on FGF, and certainly there could be some more. So that's also a contributing factor, is the stability issue of some growth factors.
Thank you, Peter. Awesome. Well, we have just a few questions left here. A question here from Kathleen. Is there a problem with tagging the proteins you make with a flag tag, for example?
Mm-hmm. Yeah, I think this is related to a previous question about this, sort of relating to kind of the regulatory, if there's any regulatory issues for using tag proteins. So flag tag in particular has been used for decades for molecular biology and cell biology purposes, right? So it's well known by biologists not to really have dramatic effects on cells, where adding flag tag proteins, or you doing experiments such as immunoprecipitation experiments or immunofluorescence experiments with blotting or labeling for a flag tag really doesn't have a major effect on the proteins themselves. So that's good. We didn't use flag tag. We use these other tags, right? So there should be more work on that, just to make sure, again, you're adding TGF-beta with DSBC tag, what's going on with the cells. So there needs to be more work there. We looked at the signaling pathway, but also are the cells morphology, is the cell morphology proper, et cetera. So we don't anticipate major problems, but certainly there's more work to be done there. And then, then back to of course, the regulatory issue, right? Which it's hard for me to comment on, but my gut is I don't think it would be an issue as long as you can establish that the tag, the growth factor with the tag that you think is maybe causing an issue, you can remove that from the media. So yeah, I hope that solves that. But yeah, for me, flag is not an issue, right? Like in molecular biology, but certainly some of the tags that we used, it'd be good to see some additional research to verify that it really is not disrupting, the cell morphology and the way they grow, the way they divide, et cetera.
Awesome, thank you for illuminating the steps ahead for additional inquiry. Last question here around whether you can elaborate on the embryonic bluegill line that you used and maybe your experience of getting relevant cell lines altogether.
I wish I could answer that. Cam Semper in our group in Calgary would definitely be the guy to ask. He's, like I said, he's working on developing a serum-free media for fish cultivation. So he'll be able to comment on that. And yeah, you can find his contact information in our paper, or you can reach out to me and I can put you in touch with him. So certainly he'll be able to illuminate you on that.
Awesome. Well, thank you so much.
You're very welcome.
Yeah, sharing this time with us. Before we wrap up today, is there any kind of last reflection or parting thoughts that you'd like to share with our scientific community?
Yeah, I just like to say that, sort of what I said at the beginning, when I first entered this field, I was really struck by the kind of diversity and just the range of backgrounds and experience people have in this industry. So keep an open mind for, collaborating with people that have different expertise, because I think to really crack this, or make this industry a success, I should put it that way, requires people from, all kinds of different scientific or non-scientific backgrounds. So have an open mind, talk with people, stretch your boundaries a little bit, and learn new things. Certainly I did during the course of this work, and I will always continue to learn new things. So yeah, I would say have an open mind. Don't feel afraid to collaborate with people or connect with people outside of your field of expertise, because I think we need to do that. It's a really multidisciplinary field, and to make this, to really make this revolution, it requires people coming at it from all different types of angles. So we need to continue to do that.
Absolutely. I just got a text from a colleague who's been watching your seminar that says, Peter Stogios for president of everything. I love those parting thoughts. Thank you so much, Peter, again, for your time. And absolutely, yes. So much resonance around this call for more multidisciplinary collaboration, for more public research funding, for more open access science, and for this kind of open-mindedness that will really be the driver to get us to where we need to be. So thank you again so much for taking the time to share your research with us. Thank you to everyone in the audience who has stayed with us, and we look forward to seeing some of you at our networking session starting at right at the 15-minute mark. So thank you so much, everyone. I hope to see you again next month for our next Science of Alt Protein seminar. Thank you. Goodbye.
Bye-bye.